I would like very much to introduce my old friend and colleague, Tom Knudsen, who spent many years at the Environmental Protection Agency as one of the leads of their computational toxicology program, and is now a adjunct professor here at Indiana University, working with us. And he's going to give us an overview of using CompuCell modeling to predict birth defects. And I will turn the floor over to Tom.
Okay, so I really appreciate being invited to speak. I speak usually every year at this CC3D workshop, and I usually present details on the latest model we developed. But this year you had a nice talk from Jörg Burkhout on neural tube defects. So I thought what I would do is talk more about applications of the modeling. So I'll run through a bunch of the models that have been developed and talk a little bit about how you might use these models and in the context of predicting birth defects. So really it's developmental toxicity, but you can deal with genetic birth defects as well. As James indicated, I was at the EPA. I retired last year before all of the chaos, and have been working with James, I think since 2009 when we first met at the first International Virtual Tissues Workshop that was organized at the EPA. And since then, we've sort of grown together in terms of looking for models that have practical application and can deal with the complexity of embryonic development in a practical way. So at the EPA, we were involved in computational toxicology research. And the goal was to develop new approach methods that could use in vitro and in silico models to predict developmental health hazards and do that with less reliance on animal testing. Two workshops that came out in parallel, or two roadmaps that came out in parallel, one from the FDA, their predictive toxicology roadmap was building areas for improved predictivity and new challenges to support replacement, reduction, and refinement, or the 3 Rs of animal studies. And the EPA's work plan, so new approach methods, prioritizing efforts and resources to reduce animal testing while protecting human health and the environment. And the links to those documents are there. There are many other documents that have been published on this topic, but these are two of the original ones. So birth defects in this country affects 1 in 33 live births. In most cases, we don't know what causes them. A number of birth defects we know are chromosomal problems, some are genetic problems, and some are caused by drugs or environmental exposures during pregnancy. But by and large, it's thought to be the interplay between genetic factors and environmental factors that are responsible for most of the birth defects. The period of time that anatomically structural defects arise would be between the 3rd and 8th week of pregnancy. In most cases, women don't know they're clinically pregnant until between the 4th and 6th week. So this is a problem that faces the sociology of the field, in that many women, for example, will stop drinking once they know they're pregnant, but they may have already done damage to their unborn. The current way that testing is done to determine a developmental hazard is through pregnant animal studies, mostly rats and rabbits. And the question that we've been wrestling with over the years is, can computational embryology reduce the need for animal testing in a reliable manner? In other words, can we incorporate computational embryology into new approach methods for predicting developmental effects? So again, the conventional way of doing this would be to look at the embryo during its vulnerable period, use information that's currently available. We know a lot about the biology of embryology, and so we can build computer models of the embryo. But typically what's done is the work from animal studies is extrapolated to make a prediction in the human. What we want to do is maybe turn that over a little bit so that we could start with a computer model of the embryo that's validated against actual biological information, and then extrapolate that through incorporation with a number of in vitro and other in silico models to predict developmental hazard in a human. So James Sharp, who is a well-known embryologist, wrote a really nice review article in Development in 2017 on computer modeling in developmental biology, growing today, essential tomorrow. And I extracted this quote from his article, which is really cool, because he says, much as a microscope is a tool to help us see things that are too small for our own eyes, computer modeling is a tool that can help reveal the dynamics and predictions of an idea that is too complex for our own brains. And so the idea here is to use the computer not only in a machine learning way, but use it as a virtual microscope to really get in there and examine the cellular dynamics as normal development is occurring, and how we can disrupt that and make predictions about key events and birth defects. Shane Hudson gave a beautiful talk yesterday where he showed just exactly how detailed you can get with a CompuCell3D model in terms of replicating palatal development and simulating cellular changes. And this is, Shane worked with us, and this is really a very telling way of trying to build novel computer models that have some practical use. At the 2009 workshop, we had a really nice talk from Richard Newman, then of a company, Endogenics, where they built this, what they called a self-regulating virtual embryo. They've got 21 cells in this particular model, and they asked the question, how are these cells regulating size? If you'll see a shark bite come out in a moment, and that'll take a group of cells away. And now the system is recomputing, and they've used an evolutionary algorithm for this particular model. And you'll see after time, it'll settle back in its original homeostatic 21 cell state. A lot of the rules that were developed by the computer did not work. Some worked too well, and you got too many cells. But in this particular combination of rules, everything was set so that they could recover the starting configuration. And these cells are computing with one another in terms of who should grow, who should die, who should maybe adhere slightly differently. So this type of artificial life is interesting because these are phenotype-based models. You start with a question, how does it get to a particular effect? In this case, how do you take a group of cells and pull them out of a model and have the system restore? So that's anatomical homeostasis. And these models are using computational logic. And very importantly, they're self-organizing systems. And I think this is something that we've really been impressed with in using CompuCell3D, is that you can build self-organizing systems that can end up reproducing the phenotypic protocols that you're interested in. The other thing that's been interesting with this approach is that they have a fair amount of intelligence. They're smart models. They can actually fill in for missing or incomplete biological information. In fact, we can focus on key molecular pathways and cellular changes and allow the CompuCell systems to evolve. And they actually, in many cases, do things that they're not encoded to do, that actually follow emergent biological properties. And we think that they're very useful for hypothesis generation. So I'm going to give you a few use cases, but you can experiment with limitless what-if perturbation scenarios and say, if I perturb this cell or this pathway, what is the impact on the phenotype? So CompuCell3D has been used to simulate a number of different birth defects. I show some of the systems that have been published or or reported to date. I'll talk about some of these. But the the idea is that what we're doing is building computer models of very critical developmental events that we know can be perturbed to cause specific birth defects. So we're not trying to build a computer model that's going to cry or that's going to spit up on you or soil a diaper, but what we're looking for is computer models that can quantitatively replicate critical events that we know are the origins of specific birth defects. So we have to understand something about key events at stages of vulnerability to particular birth defects, such as a cleft palate or a neural tube defect, as presented earlier in this meeting. Again, the anatomical self-organization is important because what we do is apply the rules that enable multicellular interactions by their physical position. And what we look for are the emergent features that can computationally decode essentially a virtual blueprint of dysmorphogenesis. And we feel that over time, progress towards a fully computable virtual embryo will build on the proof of concept case studies that I'll talk about. There's a few that are still in progress. Dr. Glazer and I are working with the team to look at intestinal development for a prenatal condition known as necrotizing enterocolitis. That's just getting started. But there's plenty of systems that can be modeled that we haven't even touched yet. I listed some of the major contributors to some of these systems down below. A lot of them are from the EPA and a lot of them are from our collaborators or other collaborators of Dr. Glazer. I took the time to sort of put a little bit of information in on what these models can show. So you don't have to read this slide, but the embryonic systems are the ones that I just showed. The relevant biological processes by their Go ontology evidence codes are shown here. But importantly, the window of time that these models are fit for purpose are shown here. So we've calibrated that to the mouse embryonic stages. Mouse, as you might know, has a 19 to 20 day gestation period, and the critical period for these models is between day 5.5 and 15.5. The corresponding human periods are from the 3rd to the 12th week post-conception. So those are all given here. The emergent features are described. The adverse outcomes for particular birth defects are described. And the CC3 model references are given as well. If we take this information and we put it sort of in a graphical form. Now, the way that FDA and EPA tests for developmental safety is by a standard guideline test, a world guideline test from the Organization of Economic Cooperation and Development. It's test number 414, which is called the Prenatal Developmental Toxicity Study. And this shows the timeline of that study against the models for the virtual embryo that I just showed. The box down below says what the guideline study is designed to do. It's given general information concerning the effects of prenatal exposure on a pregnant animal. The rat is the preferred one, and its organism. The pregnant animals are dosed with the particular drug or chemical during the embryonic period, covering from gastrulation through palatal-fusion. That's shown up on the timeline for the mouse, E5.5 through 15.5, which is the period that would be tested in guideline 414. So the way that those studies are interpreted is that near-term litters are evaluated for observable defects in growth, particular malformations, or even prenatal death. And as you can see, the suite of models that we have, while no model covers the entire period, but the suite of models pretty much covers that period of time. Now, granted, each different system may have pathways that are unique to a particular process, but the idea of forming a fully computable embryo would be to integrate everything together and make a prediction of what might happen if a chemical or a drug at a particular dose and stage of administration might be given to a pregnant human. What would be the risk based on an animal study? And we don't want to do the animal study. So the reference for that test guideline is shown. You can get to all of the OECD test guidelines for development, for cancer, for neurotoxicity, for pretty much anything from their website. So the workflow that's required to develop each individual case study is shown here. It's complex. You need to define your case study or formulate the problem. Requires a lot of literature mining to extract relevant information, to build ontology maps of what's controlling each system. Then you go to your CompuCell3D framework. You input your parameters. You start rendering synthetic prototypes through the multi-scale simulations where you can analyze the output parameters quantitatively. You can do sensitivity analysis on the model, say genetic perturbations that can be validated against real biology. We call the models generated by CompuCell3D as Cybermorphs. And we're comparing Cybermorphs against actual data to calibrate the system. And then ultimately you want to operationalize these models using stressors of different sorts. So for example, chemical stressors was of interest to the EPA, and start making quantitative prediction of what outcomes you might see. So that's sort of the general workflow. It can be applied to any tissue or any disease system. It doesn't have to be developmental, but this is what we've found over the years seems to work. So I'll go through a couple of vignettes. Not going to go through the details of model construction, but just how they can be applied. So this was the first model that we developed. It was a postdoc, Nicole Kleinstruer, who led this one, and she's actually now high up at the NIH implementing their NAMS research program. But what we did was we took the lead from work that Jim Glazer's group was doing on vascular development, showing how you get self-organization of endothelial networks in culture. We applied the signals that are known to regulate vascular development, and those signals are shown here. There's something like 7 of them. Built a CompuCell3D model using endothelial cells, macrophages, and stromal cells that are interacting through these different signals, and made some predictions. One of the things that we were interested in was thalidomide analogs, because thalidomide has been shown to cause birth defects by disrupting immature blood vessel formation. And in our data set, we had bioactivity data for thalidomide analog, 5-HPP33. And we applied perturbation data from these different signals to the computer model. And what Nicole showed was that she can make a dose-dependent prediction of effects on disruption of vascular development from 5 micromolar up to 40 micromolar, where she found complete disorganization. The Japanese group that developed 5-HPP33 published only a very high dose where it pretty much eliminated everything. And Nicole wrote to them and asked if they had some pictures at these different dose levels, and this is what they sent on the bottom. And as you could see, the computer model not only predicted the effect right, but it got the concentration right. And that was really a big surprise for us, and actually really an encouraging feature that led us to look at more complex systems, such as limb bud outgrowth. And as I indicated, disruption of vascular development has been associated with thalidomide-induced phocomelia. This model is one of my favorite models for a number of reasons, but we built the core control signaling network shown up in the upper right, which is an interaction between different cell types in the limb, different positions in the limb as it outgrows, and the different signals that are exchanged between those. So this is captured from the literature. Down on the bottom, those cell fields are incorporated based on in situ hybridization patterns into CompuCell3D, and that network is essentially put into motion using CompuCell3D, and you can see the time-dependent evolution of the predicted signal gradients that are expressed in the limb. So we could test this in a number of ways. So one thing that we did was we used 5-fluorouracil, and we used data from our data set that tested 5-fluorouracil in embryonic stem cell cultures at different concentrations shown in these panels. And the interesting thing is, so you can invoke a concentration-dependent cell death, which is what fluorouracil does. It also inhibits cell proliferation. And as you can see from the panels, you can make predictions of effects based on how much cell death is produced and what potential effects they might have on polarized outgrowth of the limb bud. And as you look at this, one of the things that we were focused on was the impact on the sonic hedgehog lineage. So you can perturb that model right around the time that just before sonic hedgehog is expressed, and a lot of the dead cells that you see in red are actually affecting the part of the limb where sonic hedgehog is expressed. And sonic hedgehog is a major patterning signal in early limb development that forms that paddle-shaped limb bud. And so you could begin to quantitate the impact of that disruption on sonic hedgehog cell counts, so cells that are expressing sonic hedgehog. And interestingly, so on that graph, the black line shows the time-dependent growth of those cells in a normal system. If you perturb that with different amounts of 5-fluorouracil in a synthetic model, you could see that you're actually getting waves of disruption of cell growth patterns. So those sort of cyclical recovery stages are dose-dependent, and that's actually something that has been reported in the literature. Shane talked about this model yesterday. I won't go into much detail on it, but I wanted to highlight a few things that he said and a few things that he didn't get to. But in the case of the palate, of course, in the embryo, the right and left side, if you're looking at the roof of the mouth of the embryo, have these two shells that have to come together and fuse together in a controlled way. And as Shane had shown in the model that he built while he was working with us, that breakdown of that medial edge seam is a critical event. If that doesn't occur, then as the skull continues to grow, it's going to pull those two shelves apart. The signals that Shane incorporated into the model are shown to the right, at least some of the signals. And the phenotypes from the different knockouts, virtual knockouts of those signals are shown to the right. I want to particularly draw your attention to the TGF-β3 EGF signals on the bottom. Shane talked about those in some detail. One of the things that he didn't say was that, so when we went to publish this paper, one of the reviewers came back and said, I've been working on the palate for 20 years and I've never seen a phenotype as shown in figure 5. That's the TGF-β3 knockout figure. And what Shane found is that there was actually a human study published by Dudas et al. Shown in histology images that actually did show that if you knock out the receptor or look at a mutant receptor to TGF-β in the palate, and these are human palate shells, and put them together in culture, you get abnormal expansion of that medial edge seam. So in fact, we didn't code that into the model. That was sort of what emerged from one of these analyses, and even an expert in the field questioned that result. But after seeing our response, he went right ahead and said, okay, the paper's accepted. The other thing that I wanted to highlight with this model, so he showed the network that was used, and he indicated that that fusion process is the result of a bistable switch between TGF-β3 and EGF. And that switch can be disrupted. He talked about disrupting it by dioxin, but it could also be disrupted by disrupting retinoic acid gradients during development. And the interesting thing with this switch is that you can model different amounts of hysteresis in the model, as he showed. If you have a tight hysteresis model versus a loose hysteresis model, you see very different patterns. In that tight switch, you see both of them originally have a delay in medial edge breakdown. But if you look at the difference between the switch, the tight switch, actually, it was a reversible change, whereas with the loose switch, it was a permanent change. So the dynamics in these switches can be played with and hacked in a virtual palate, and you could start getting some predictions on what relative amounts of switch dynamics might be critical for a particular process. That becomes important when you want to apply that information. So if we go back to our ToxCast database for chemical caftan that's known to produce cleft-palate, at least in rabbits, and we looked at our in vitro assays for EGF signaling and TGF signaling, we found that caftan was increasing EGF in a concentration-dependent manner and decreasing TGF in a concentration-dependent manner. So it's essentially, you could take these two pieces of independent data and put them together and envision a switch, an EGF-TGF switch. And then we can model that switch in the virtual palette. So I'm going to show on the bottom, that's an automated concentration response where you could see the critical effect at a particular concentration where I've indicated in the dotted line. And that makes a prediction of what might be the level of the switch dynamics that would be critical for this chemically induced model of cleft-palate. I won't show that data, but we've actually used reverse dosimetry to go back and correlate that to what is responsible for producing that in a pregnant rabbit, and it came actually pretty close. Another model where fusion is important is urethral-closure. The scanning EM shown on the left is the genital tubercle of the embryo. This is the human embryo. Embryos, at first when they develop, they're sexually indifferent. And at a certain time in development, when the testis starts to be organized and produce androgens, it triggers growth of that genital tubercle, and you end up having elongation and folding to create the closed urethra of the penis. So Max Lung worked on this model, developing it in CompuCell3D. He came up with a schematic to evaluate the urethral closure, and that yellow urethra tube is initially opened in the model, and it's surrounded by mesenchyme that's sensitive to androgen triggering. And the signaling network that controls this is shown in the middle. It's both sonic hedgehog and FGF signaling, as well as androgen dependent. So the models are shown up, the model is shown up in the right, and you could track the expression of those two signals. And what they do is they cause some programmed cell death to occur that allows those fusion events to take place, and you get a closed urethra. So we can go in and we can use a compound that's known to disrupt androgen signaling, so vinclozolin, and go back to our database and ask what question do you start, or what concentration do you start being able to disrupt that closure process. So if you think about 100% androgen as a male genital tubercle, or 0% androgen as a female genital tubercle, you can actually show that the model can show that you get different degrees of closure. You can run these models dozens of times and come up with statistical probabilities of closure and find out what the biologically relevant reduction of androgen signaling is. And that turned out to be somewhere in that range, where about a 30%, maybe to 60% drop was able to predict statistical effects on the incidence of urethral closure. Earl Gray's lab at the EPA actually did that experiment in rats, and they reported for different phthalates that the biological relevant reduction in androgen signaling was somewhere between 40 and 50%. So the model, again, did a pretty good job predicting what was actually shown experimentally. I'm not going to harp on this model, but Job had presented this yesterday, so some of you may not have seen his talk, but this is another example of controlled fusion events forming the neural tube. Failure of that to occur results in severe birth defects. This one that was being modeled was spina bifida. And what Job was working on was a control network that we had developed a few years ago. And he sort of modeled what the key events might be for that process of neural fold elevation, remodeling through different hinge points. So there's a couple of hinge points in there, and those hinge points drive the closure along with different growth kinetics in the ectoderm and mesenchyme. And what Job was able to show is that you could knock out different levels of these signals that are controlling the process, shown on the lower right, and produce phenotypes, complete open neural tube defects, or neural tube defects where the skin closes, but the neural tube doesn't, sort of called a closed neural tube defect, or midline fusion defect. And he looked at incidences of these occurrences in the literature, mostly in mouse models, and found that his simulation actually anatomically reproduced pretty closely what you see in different knockout models. But with this, you can actually control the amount of knockout of each particular gene. You can actually overexpress these signals. You can look at combinatorial effects of perturbations and make quantitative predictions of what percentage of each phenotype would be predicted. And that's shown on the lower right there. The model I presented last year on gastrulation. This is an important process. It starts very early in development. Remember the timeline I showed earlier. The epiblast is a pancake-shaped structure on the 5th day of mouse gestation, the 3rd week of human gestation. And the hallmark feature of this is the primitive streak. I know Dr. Glaser and his colleagues modeled primitive streak formation in 2010. What we were interested in here was not only in primitive streak formation, but in trying to gauge the dynamics of mesoderm specification, because that's really what the important aspect output of gastrulation is. It's a spatial patterning and signaling that results in mesenchyme or mesoderm that's destined for different parts of the embryo. And there's a couple of important points. I'm not going to harp on the details, but a couple of important points. Number one, cells stream towards the future posterior part of the embryo. And this actually establishes the anterior, posterior, or head to tail axis of an embryo. And as cells stream, they sink in at that point and form the primitive streak. It's not a structure in itself. It's actually a defect in the ectoderm of that epiblast as the mesenchyme sinks in and migrates between the ectoderm and endoderm layers. And cell position is important here, because when and where one of these epiblast cells, which are known as epiblast stem cells, they're pluripotent cells, where they enter that primitive streak is known to be a key determinant of mesodermal fate. So different mesodermal regions destined for different parts of the embryo have actually their origins at this stage in development. And as those cells pass through the primitive streak, they become determined in terms of their homeobox specification address. And I'll talk about that in a second. So the first model here that I'll show is a 3-dimensional CC3D model. Actually, I think CompuCell3D should be called CompuCell 2.5D. Once you move to 3D, we've really uncovered a lot of difficulties, although they can be overcome. And we've worked with Dr. Glaser's group in overcoming some of those difficulties. But as you get to these complex models in a 3-dimensional context, the challenges and the cost, the computational cost, become pretty expensive. So this is a one-shot simulation. And what's going to happen is the cells are going to move to the primitive streak from the ectoderm. They're going to sink in under the ectoderm and migrate to different positions. Now, one of the limitations of this model is that that light blue rim is a physical boundary. In the real embryo, the whole system is still growing and there's no real physical boundary. Cells would move into different parts of the extraembryonic membranes as well as different parts of the embryo. But what we've done is, as mesoderm is formed, we colored them in a particular code for different parts of the mesoderm. And then what Richard Spencer did was he actually made this model transparent with respect to the ectoderm, so you can kind of see all of those dynamics. You can actually zoom in at that position of the primitive streak and see exactly what's going on. But the interesting thing is that the model was tuned here to replicate the distribution of mesoderm in a mesodermal fate map of a real mouse embryo published by McDowell et al. In Cell. And so we calibrated the patterns of mesodermal specification based on exactly what those investigators showed in their fate mapping of the mouse embryo. Before I leave this slide, just to point out, famous embryologist Louis Wolpert once said, "It's not birth, marriage, or death, but gastrulation, which is truly the most important time in your life." So you could take that model, you could do lots of things with it. You can make these arrays where you focus on the key signals in the system, from electronic knockouts at different levels to the isomorphs, which is the normal, to actually increase signals, hypermorphs, and start quantitating cell outputs destined for different parts of the mesoderm to try to make inferences on which particular growth factors might be most important in specifying the system. And what we found was actually fibroblast growth factor 8 induced cell migration was the biggest limitation of mesodermal formation. But if you intervene with that process, then you begin to alter the molecular address of those mesodermal cells. So when they get to their position in the embryo, if they don't have the right address, they don't necessarily know what to do to contribute to, say, an eye or a heart or a blood cell or a limb mesenchyme or a palate. So the specification of those cells is important, although what this map is showing is the numbers of cells destined for each region. But we can incorporate that homeobox code into these models. So you may not know much about this, and I'm not going to change that, but the Hox clock is a critical aspect of early development. And the homeobox genes are represented in mammals as 4 clusters of up to 13 paralogs that are expressed in a developmental and positional information. So that's what's called collinearity. So you go from the low numbers or the 3' side to the 5' side in order of development and in order of anterior posterior positioning. And cells are actually measuring time in the epiblast by the epiblast and its derivatives by the activation of the combination of 39 homeobox genes. And the address of each cell is dependent on the pattern of these genes that's expressed. And as I indicated, as those cells migrate through the primitive streak, that's where the homeobox state is actually set. And there's an autonomous clock in the embryo, which we've incorporated into CompuCell3D. And the central control of that clock is through the genes, the caudal-related homeobox genes, CDX 1, 2, and 4. Those are the central regulators of the clock. And some of the key signals that affect the rate of that clock are retinoic acid and fibroblast growth factor. So now, instead of coloring the epiblast-derived mesoderm by their position, we've colored them by homeobox fate. Now, we should have done all 13 paralogs, but for simplicity, we just did 4 paralogs. And so it starts out, you'll see the anterior paralogs in light blue, the green paralogs, next the yellow paralogs, and finally the posterior or red paralogs. So now we can start messing with the signals that regulate CDX. And if you underserve that signal, so if you look at the map to the left, the anterior genes are largely retinoid responsive, posterior genes are largely FGF responsive. CDX is coordinating that. If we do things that reduce the expression of CDX, so you see this hypomorph, and what you're going to see is that you don't get the progressive activation of those more posterior colors. So it'll, in this case, it's an extreme case. They're all going to stay in the light blue. So all of those mesoderm cells that come from that model, even though they're destined to different parts of the embryo, such as the posterior region, they're going to carry an anterior address with them. Or you can go the other way and overexpress CDX, and you'll see the opposite condition here now, where you have too much or too rapid a change. And so what's going to happen is that all the cells that are forming may have a posterior address, even though they might be destined for anterior positions. So, you know, these are things, I'm not trying to do anything more than indicate what you could do with a CompuCell3D model that it may be difficult to do otherwise. And so with this type of segment identity model, we can start asking questions about how chemicals may be affecting the fate of different mesodermal cells. So, for example, on the left, uncontrolled retinoid signaling during gastrulation is going to anteriorize the system. It's going to shift your posterior segment identities more towards the anterior. Or on the other side, uncontrolled fibroblast growth factor signaling is going to posteriorize the system and shift anterior segment identity more caudally. So in either case, you're going to get maybe some aspects that anatomically are normal, but they're carrying the wrong address. So functionally, those cells are going to be confused and may not respond in their appropriate way to whatever signals in their embryonic positions are given to them. Okay, so just now to sort of wrap up. So what can a virtual embryo portfolio bring to risk science? So I've listed a number of key elements that really are lessons learned from executable models in CC3D. We always keep in mind that our computational unit and biological unit are the cell, and that's the agent of these models. And what we're looking at is their dynamics in the computer in response to different types of alterations or perturbations. So CompuCell3D treats individual cells as autonomous agents. So each cell is its own model, but they're in a shared microenvironment. And the coding specifies individual behaviors, regulatory signals, as well as biophysical properties of the model. But then time-evolved simulation is enabled by how that biology unfolds in an artificial life-type model. As I've indicated, we've seen plenty of examples where the model can fill in for missing or incomplete information. So they're tuned by phenotypic protocols. And so there is a certain amount of computational intelligence that's involved. So they are smart models, or they can be. Some of them are pretty stupid. And what we can do is we can run them many times. So they are stochastic, and we can get then a probabilistic rendering of how a molecular target results in an incidence of an adverse outcome or a cybermorph. As I've indicated, they're amenable to different ways of automated calibration, parameterization, and as well as uncertainty analysis. So those that are familiar with uncertainty analysis would know that there's statistical ways of knowing how accurate your model or how reliable your model is, and each uncertainty at each stage amplifies as the models progress. But you can do limitless what-if scenarios. You can determine what the criticality of the system is. Where are the critical weaknesses or critical phenomena that might set a system off in the direction of a birth defect? And through that, you can start generating different hypotheses on what those mechanisms might be. And I got this phrase from Dr. Glaser, but really a fully computable code-to-dynamic simulation of human variability, life stage, and disease process is, I think, what the bottom line of the advantages are for this model. So there's really two ways of thinking about criticality in terms of a translational way. And I would encourage you to think about building models with these two things in mind. So you can start with forward engineering. So suppose we know what a particular molecular mechanism or perturbation might be. You can ask the question, how far can CC3D take us to hypothesizing a particular phenotype? So sort of mechanistic models from induction to elicitation. But you can also reverse engineer. So you can start with a phenotype. Suppose we know a phenotype, a birth defect or a particular disease. Then ask the question, how far can CC3D take you to inferring what that molecular mechanism might be? This is commonly done right now by machine learning, looking for molecular changes that are connected to particular phenotypes. But if you think about a specific system and you want to work backwards to the molecular level, so those types of machine learning predictions can be incorporated into these models to determine the veracity of how well those predict a phenotype. So I'm going to leave it at that point. I did not have any exercises to use for this particular lecture, but I do have a slide on food for thought, how you might be thinking about implementation of virtual tissue models. So in time remaining, I'm certainly happy to answer any questions, or if you want to think more about the system from a translational perspective, from a basic science investigational perspective, or even an operational perspective for implementation. How do you sort of think about models in that context? But I think one of the key things, and this is a lesson that we've learned at the EPA over and over, is that communication is really important. You can run proof of principle for anything you want, but really to convince users, regulators to think about your models, they have to have a certain level of comfort that these models are actually telling you something. And particularly if you're trying to replace animal testing, which is the gold standard, they have to be shown to be ready for use. And I think this is someplace that we really need to do a much better job in terms of communicating these models. One thing I found is that risk assessors, they can look at many graphs, they can look at tables, they can look at heat maps, they can look at things that they don't really understand. But when you show them anatomy, they understand anatomy, and they can see something happening that might be relevant to what they're thinking. And that's really, I think, a good way of implementing these models in a more translational way. So James, I'm going to leave it there. I'll leave this slide up unless anybody has any questions to go back, and happy to chat and do whatever you'd like.
Great. There are some questions. Thank you so much. There are some questions in the chat. Can you see the questions, or should I read them out to you? If you do read the questions from the chat, please read the name of the person who asked and then read back the question before you answer.
It'd be better to do it yourself, because if
okay, it's hard for you to visualize it. Okay, so the first question is from Rodrigo. You have many different parameters in these network models. I'm wondering if they're identifiable from data. How do you establish physiologically relevant concentrations of morphogens?
So the true answer, Rodrigo, is it's trial and error. There are some studies that actually have looked at spatial distribution of morphogens, but they're few and far between. So the way we do it is again, it's a phenotype-based protocol. So we say what concentrations tune the model to give the right anatomy and the right morphology, the right spatial and temporal behaviors.
There's a comment in the chat. It's gradual. Okay, so the second question is from Clara. How far can models really replace animal testing so far?
So far? All right, so when you're dealing with an animal model for pregnancy, for example, you not only have the effects to the embryo, but you have the effects to the mother as well. Obviously, those are not explicitly expressed in these CompuCell models. We have to infer a lot, make some assumptions from what might be happening to the mother and what that's doing to the placenta and to the embryo. So it's a very good question. I think that's going to be one of the limiting factors in communicating and selling this to translators, or to risk assessors, because they don't care necessarily about the details. They just want to know if they produce, or if the pregnant animal is exposed, what is going to be the impact on development? Whether it's maternal, placental, or direct effects on the embryo may not matter so much. Now, that said, one of the NAMS approaches that's actually very good at predicting developmental toxicology is the embryonic stem cell model. That can predict developmental toxicity with a confidence of 80 to 85%. What it does is it misses a lot of things that an animal study picks up, which could be because of those maternal effects. Now, the other thing is that in an animal study, you've still got uncertainty in extrapolating, say, a rat study to a human, or a rabbit study to a human, or even a zebrafish study to an animal model. It's about 60%. So rat predicts about 60% of a rabbit, zebrafish predicts about 60% of a mouse, and you would think if you put them all together, you're going to get 100% prediction for humans. But actually, that uncertainty multiplies, and you get less certain as you start necessarily just adding things together. So in the field, we know that zebrafish can pick up a developmental effect, a developmental hazard, but whether or not that's a direct effect on a mammalian embryo is not certain, because the zebrafish doesn't have the placenta or pregnant mother. Now we know that the rat can pick up a lot of potential developmental hazards to humans, but not everything. And the rabbit can pick up things like thalidomide that the rat was not able to pick up. So, you know, there's a lot of value in animal studies. They work. It's not that they don't work. The problem is that they're very expensive. They're timely. Takes a long time to do them, and it uses a lot of animal lives. And those are the things that we're trying to get around by using information from human cell-based assays, and then using computer models that translate that information into a potential human relevant phenotype. So it's a very good question, but in a long answer, but that's where we are.
Okay. Luis asks, do you have any specific references for machine learning approaches that you think would be useful, relative to these slides?
I'm not sure what you mean by preferred. You mean types of machine learning algorithms, or?
Are there any specific references you'd have people start out with that are relevant to the slides where you talked a little bit about machine learning?
Yeah, there's been quite a bit, actually. So machine learning tools have started with chemical structure information. They start with kinetic information. They start with biological information. There's many types of machine learning approaches that have been used. I'd like to see CompuCell3D use machine learning to write the code, actually. I think that would help.
We're working on that.
Yeah, okay.
If somebody wanted to help us do that, we'd love to have it. VS Code, of course, or if you use VS Code or Visual Studio, they have a built-in coding assistant. Mm-hmm. But you would like to have the coding assistant trained specifically on CompuCell. At the moment, if you use ChatGPT to generate CompuCell code, it does a pretty good job, but it makes some mistakes. Mm-hmm. In particular, it doesn't know the difference between CompuCell3 and CompuCell4, so it uses old-style code. Uh-huh. And so the Morpheus package, they've been working to train an LLM to generate Morpheus XML from Morpheus. So that's still in process, but works pretty well. So this year, we're not quite ready to start teaching LLMs for coding, but I encourage you to, once you know the basics, you have to know the basics yourself, but once you know the basics, I encourage you to explore using LLMs to do coding. And if you're using something like, depends on your ID, but if you're using something like Visual Studio, it can help a lot. But we would definitely like to be more sophisticated. The other place that LLMs can help a lot is in annotation. in principle, we didn't talk about this the other day, but in principle, when you're creating, especially if you're going to use something in a regulatory medical context, you should be able to track back, where does the parameter come from? What does this parameter mean? And NIH has put a lot of work into creating ontologies that give specific definitions of what a cell type is, what a behavior and process are, what a molecular species are. And in principle, if you want to make reproducible models that are usable in a regulatory context, every concept in your model should be annotated from one of those reference ontologies. At the moment, doing that's quite time-consuming. And that's a place where LLMs might be able to help a lot.
You know, one thing as the editor of a journal, scientific journal, that I'm finding is a concern with machine learning approaches coming from certain countries that they're not using scientific method. They're just sort of haphazardly looking to see, oh, this point is, this parameter is connected to that parameter. I think that use of the scientific method should not be lost as we're sort of building these types of models and approaches.
I would agree completely. I don't think LLMs replace us. I think they can make us more efficient. And again, I would use the example of translation. Mm-hmm. If I type English text into an LLM like ChatGPT or Claude, and I ask it to translate it into French, if I don't, it will do a decent job, but it'll make some mistakes. If I speak no French at all I can do is use it. And if it's wrong, then somebody who speaks French will tell me I made a mistake. If I know some French, I can catch big errors, but I may not be able to fix the problem, but I may at least know their problems. If I know French very well, then I can fix the errors myself. And so it's a little bit like that with coding. For somebody who doesn't know any coding, these systems are generally not that useful because they still make some mistakes and you don't know enough to be able to catch it. And so just as with the language, the more you know of the language yourself, the more efficiently you can use these tools. But it doesn't get you off the hook. What it does do is if you don't remember what is the syntax for the such and such function, that it'll pick up for you. And there are certain kinds of things that it does quite effectively. I want to iterate over all neighbors of a cell in CompuCell. How do I do that? In the old days, you'd look it up in the manual. You might look it up in Reddit or someplace else. Now, those kinds of things are pretty well doable with the LLMs. And so I think we're still learning how to use these well. And I agree with Tom that this doesn't get us off the hook for understanding what we're doing. Although some of the new LLMs that have scientific reasoning are pretty impressive that way. So it's interesting. And that's something where I think the younger people probably are going to be the ones who make the big advances. Tom and I are a little set in our ways, perhaps. And so we're counting on you to tell us how to do things. There's a question here, which I think is a very visionary one, and it's something that I've wanted to see happen for a long time. And I'd love to see somebody work on it. And we actually have a little bit of tools for it, but not enough. Would it be possible to go from videos of experiments to actually define the cell properties and behaviors in CompuCell? In other words, to infer the properties and behaviors from the observations.
That's a question for me. So, you know, I think that's important. The one criticism, of course, as a morphologist is, to me, all of these models, no matter how sophisticated they look, they're still Pac-Man models. And what I'd like to do, or I'd like to see, is that we translate these videos into photorealistic simulations. So we were working with a group at University of Illinois to actually do that until that group sort of disbanded. But if you think about, you see it all the time in planetary science. So the graphic engineering that goes into some of these planetary videos are just phenomenal. And if you could just imagine how we could use that biologically to make our virtual microscope Pac-Man type images look more like the real thing, I think that would have a lot of impact. I mean, it may not be necessary for risk assessment, but I think it's important for communication and validation. I don't know. What do you think about that?
I think it's very inspirational. I don't know who you were working with at Urbana, but of course, Zaida Schulten, of course, has done a lot of work in that area at the molecular dynamics level, among others. I think, again, that's the kind of thing where the people in this room can take that forward and make it happen.
It's just this slide. You know, this is kind of what we're getting at. You know, I think at the stage of working with young folks, you know, where the passion lies, don't stand idle and watch technology race by. So, you know, you really need to think about, you don't want to be the guy looking like he thinks he's a hoop snake watching the field go by, that this was a really cool cartoon. So, you know, that was a thing. And it's important, you know, that you get involved with more of these global programs that are working on this. So just in terms of, you know, trying to instill some inspiration as a parting thought, we think you're the right people to move this field forward, and we're here to help.
I just was putting in the chat that I think some of Tom's examples, that vasculogenesis simulation, that limb bud simulation, I don't know if there's old code available for it. Getting Shane Hudson's simulation, which doesn't run in current CompuCell, running again. Implementing those, documenting them, putting them up in the CompuCell model repository as working models could be a really good learning experience for some people. And it would be achievable in 3 days in a hackathon. You'd learn a lot by doing that.
All right. Well, thanks for your attention, and thanks for the invitation to speak, James.